Good morning, everyone, and thank you for joining this webinar. It's going to be an exciting time in which I can go through, hopefully, a little bit of the overview of exome analysis in general, and as well as going through some analysis of my own exome and the implications thereof. So it looks like we're all getting going, and as people are continuing to roll in, I wanted to do a little bit of shopkeeping and let you know about the questions pane on the right side of that webinar doc. I'm going to actually pack out this presentation. It's going to easily take up an hour, but I definitely do enjoy spending time at the end answering people's questions as they come up. So feel free to put them in now, and I'll kind of go through them as much as I can. If I don't get to them, don't feel offended or anything. I enjoy following up outside of the offline or outside of this webinar forum. You can get me on Twitter at @GabeInformatics or follow up by email. Certainly we'll be covering a lot of topics, and many of them warrant an ongoing discussion. Okay, so let's get started and talk a little bit about what we're going to cover today. So to start with, I want to really give a high-level overview of exome sequencing and its progression from the research to the clinical setting, and now as we talk about direct-to-consumer genomics. It's a very exciting progress, and there's a lot of success stories to cover, and I certainly don't intend to be comprehensive, but it does set the stage for the type of analysis and what I intend to do with it today. So the big question then as we talk about this progression is, what will the impact be of direct-to-consumer next-generation sequencing, and really the progression of consumers being their sort of advocate of their genomic data and health data in general. So it's a very interesting discussion there I want to touch on. But basically, I am going to focus on just my own exome today. And what I intend to do out of that, I'll be covering as well, and the types of things I expect to be able to learn, and more sort of comprehensively the things I don't expect to be able to learn. That should take a little bit of time, but really we're going to spend a lot of time going through the actual analysis today. We're going to spend time actually doing, not interactively, but by sort of way of discussion and then through validation, the actual filtering, annotation, and interpretation of my own exome, and the summary of some of those results. Okay, so let's start then with that overview of how exome sequencing has fared in the last couple years, because it really has only been a couple years since it's gotten to the point where it's been prolific and affordable and consistent in its performance. And the papers that have come out of labs such as, the original ones on the Kabuki syndrome, I know in 2010 or 11, we also had Dr. Golson Lyon down in Utah doing it's actually an X exon capture kit, but discovery of a novel disorder and finding and diagnosis there. That's a webcast that I actually gave a month or two ago about reproducing those results. So I've definitely touched on this type of information before. But what I think is a very representative of this success story, but also sort of representative of the technology capabilities and limitations, is this publication recently by Dr. David Goldstein's group at Duke University. And what he wanted to do was essentially take a bunch of patients that had a huge diagnostic odyssey with no final nailed down diagnosis for what was clearly a genetic disorder, and he wanted to not necessarily take them that all matched a specific phenotype or syndrome, but basically just take these lost causes and see what exome sequencing can do in general. And so out of 12 individual patients, and they all contained both the patients and their parents, so they were done in a trio analysis setting, 6 of them got to known diagnoses and 1 with a sort of a potential diagnosis. And that may seem like a 50% hit rate, but again, think about the fact that these were individuals that had huge long histories of going to array CGH and other types of genetic tests and probably many different gene panels without finding a result, and exome sequencing got to a very clear, functionally validated diagnosis. And in fact, one of these children, this one with the NGLY1 gene mutation, that diagnostic story I was able to hear a little more from a personal level, because the father of that child was a professor of mine at graduate school, and he wrote a very poignant and articulated blog called Hunting Down My Son's Killer, that's got a particular widespread popularity on the internet. But it is actually an interesting example of someone who's a father who's very passionate about trying to help his undiagnosed son come to a diagnosis and potentially a cure, driving that progress towards a diagnosis and then now onwards. He became knowledgeable in molecular genetics, and I talked to him and he understood perfectly everything that was going on at the sequence level that was happening at the lab. There was actually some hiccups where they found one gene that they thought it was there, and then they found the final gene that got the functional validation. And so it's a great example of the fact that in this day and age where individuals are going to be spending their whole life with their genome and their diseases, they are sometimes the ones to really drive forward the diagnostics and at least be able to become experts in their own individualized issues when they are individualized in the case of this novel disorder. And so I'm going to talk about that more when we talk about consumer genomics in a second. But the point of this overview here is that I think this paper represents this sweet spot of exome sequencing, and it was done in a research setting, but I know they're transitioning to implementing a clinical exome sequence at Duke as well as many other institutions, including Baylor College of Medicine, etc. But essentially, if you have these conditions met in your experiment or in your research or in your clinical case, you have a rare disorder, so it's not just something that has a high population frequency. It's congenital, so you really have a strong genetic hypothesis that this was not environmental or otherwise post, other non-genetic causes that can definitely attribute to very distinct phenotypes, and that it's highly penetrant, and that's more of a hypothesis, but you do expect that if you find a driver mutation in a gene, that same mutation would have a very similar, if not 100% efficacy or pathogenesis in a different individual. So you also expect that anybody who inherits that variant would inherit the disorder. And it's presumed monogenic, where that single mutation that is highly penetrant also essentially through its mutation of the gene is the genetic pathogenesis as understood and makes sense in context of the phenotype. So all those conditions are really a strong sweet spot, and there's plenty of research and there's plenty of papers and there's plenty of probably clinical cases at this point that follow that. And I think the next wave that we're seeing now, and certainly when I went to ASHG this year, it was exomes, exomes, exomes. Everyone had exomes, trying them out on everything. They're very affordable, and the analysis and the interpretation is starting to become very compact, as I'll be demonstrating today. The next thing then is to sort of look outside of any one of these. So looking at potentially more common variants, looking at later onset disorders, looking at things that are potentially not purely highly penetrant and polygenic diseases. And so I know there are people who are doing intellectual disability and autism with exomes and looking for individual cases, but still using exomes to try to find a genetic basis of those disorders or the genetic basis of the individualized disorder as it occurs in a single case. So those are very interesting and ongoing researches, and it's still, I think, too early to call whether these sweet spots are going to be expanded into more individual cases or scenarios. And finally, on the common disorder side, you have large cohorts being collected, such as the National Heart, Lung, and Blood Institute, who's got 6,500 exomes with adults with very extreme phenotypes, and are looking at a number of different disorders, such as they have extreme metabolites, maybe high blood pressure, and they're looking at diabetes, and they're looking at all these things, and they're looking at the power of exomes to really allow for polygenic complex disorder research. And that again is going to be something that's going to be interesting to follow, but that's at the research level. At the clinical level, I think it's really being implemented now as this end of a diagnostic odyssey, and even for those clinicians or physicians who want a shortcut, or really just, I don't want to screw around with 5 different gene tests. I don't know which exact thing it is. This is a relatively unique phenotype. Let's just go straight to exome sequencing. And the payers are starting to understand the cost benefit of this as well. So I think this is really being implemented, and we'll see today that the analytical solutions at the software side are becoming robust and transparent, and the challenges are going to remain in those cases where you find variants that have no known pathogenicity status and the complexity of the interpretation of those. And we'll talk about that a little bit today as well. So finally, looking real forward, I've been reading this great book recently called The Creative Destruction of Medicine, and it's covers a wide range of topics from the perspective of a physician who's really looking forward to the capabilities of all of our technology to essentially radically improve patient life, whether that's through patient-driven enthusiasm or their own managing of their own disorders through all these technologies for measuring your blood glucose level and your sleep cycles and your body fat and the number of steps you take, to the more complex things like genomics. And we'll be talking about that today because that's really kind of, there is this huge promised land that people are looking for, and whether it's going to be driven by the healthcare industry or consumer enthusiasts and patients with a very motivated cause is still to be seen. But this is going to be an interesting and very exciting time in which to watch this progression. So if we start talking about consumer genomics, there's really no way not to mention 23andMe. And there's definitely other direct-to-consumer genomic companies out there, such as Navigenics and DecodeMe, but I think 23andMe sort of wins it all in terms of getting sort of the both positive and negative publicity at the national scale. Not necessarily negative publicity, but they have, because they've been so prolific and been so successful, really engendered the debate of whether direct-to-consumer genomics is, one, a good idea, or two, has risks involved with it that should be regulated. And that's sort of an important distinction, is that this is data that ends up into an individual's hands. It does not go into a medical record. It does not go into a patient file. It cannot be used by insurance companies. There's already federal regulations in the United States to prevent discrimination based on genetic data, at least at the insurance level, not necessarily at the life insurance level, but at the healthcare level. And again, this data is not public. And at the most part, this seems to be catching on as both a good thing and very encouraged amongst people who are both curious and driving their own care as they might face something like cancer or other serious genetic disorders in their family. And so it's also important to cover what 23andMe does, because I've had some questions about, you're doing your exomes, are you going to find something about, Alzheimer's? Well, I already know all that stuff because I've had 23andMe providing their standard service to myself, and in fact to my wife and my son. I have a second son now as well, but we have our full genotype data at that consumer level that they provide, and that data is based on these common SNPs, and there's about 1 million, I think, in their chip. It may be a little bit less. It's not necessarily more is better. You want to have those variants, those SNPs, those common polymorphisms that are both well annotated and highly representative of sort of the population. But these common SNPs have been used in the past decade or so as the basis for most of these large consortiums doing this disease and trait research and associating those diseases and traits with SNPs. So all that great publication information that's out there is the key to what 23andMe is really mining that data and collating it to be a consumer level report. And it contains not only things like carrier status, and those can be quite critical, things like cystic fibrosis and even some BRCA mutations in the breast cancer, oncogene. But it also contains traits, from the more benign things like freckling and hair thickness to the more, relevant from a physician perspective, things like blood glucose and LDL cholesterol level propensities based on your genomics, and those are some well annotated things. You also have some pharmacogenomic responses, and this is extremely actionable information. Things like your warfarin dosage, sensitivity, your statin response, fun things like your caffeine metabolism. No surprises for the meet me there. I'm a very enthusiast of said drug, and various efficacy and toxicity, reports on different drugs, and that again is very actionable based on all this literature. And then finally, the heavy hitters around those 119 disease risk predictions, and those are vary from individualized cancers, things like kidney cancer versus prostate cancer, to celiac disease, Parkinson's disease, and even Alzheimer's disease. And it's important to note that they are very, I would say they're going about this as ethically and as consciously as they can, probably more so because they're a company in which they have to face all this public scrutiny, but they're doing things like locking these reports until you read information about, if you're going to look at your Alzheimer's disease report, you should know specifically all the details of the studies that provide that information, how confident they are that those reports are providing, what percent of the heritability of the genetic risk is going to be attributed to genetics versus environment, how big were the studies, how many reproductions of the studies were there. So all that information is there, and as you unlock the report, you also have links out to genetic counselors, etc. So this is very, I think this is very above board and very exciting to have all this information. Again, my family does not have a very interesting genetic history, or rather very sort of uneventful genetic history. I think my largest risk as reported to me by 23andMe is a 3-fold risk of age-related macular degeneration. That is actually something that I plan to track very closely. There are some lifestyle changes, and I'll be watching the research as that unfolds to make sure I can do everything I can to help prevent that type of age-related blindness. But that is, again, very actionable information that's in there. Okay, so great overview of that. That's great. But what the heck am I doing with an exome? What is 23andMe doing giving people exomes? What do we expect to get out of that? And that's the real big question here is, these known SNPs, these common variants that have a lot of research essentially saying this variant is associated with this odds ratio at this probability in this study, is all well and good. But what happens when you see a variant that nobody has, that's in a gene that is potentially associated with a complex or even one that's associated with a Mendelian disease? How serious should you be taking that data? How likely is it that a consumer is able to do the analysis to interpret that data? How likely is it that even 23andMe could package this data in a meaningful form? And that, I think, is the big question and why they put together a pilot program. And so their pilot program was extended only to users of their existing software because they were using their existing platform to both do QA and to kind of keep people in the fold here. And I was lucky enough to get myself and my full trio in here. And again, people may ask, well, what are you going to do with a whole trio? Well, one, this is very interesting to me. It's obviously my own data. It's my family's data, and I want to track things like on my wife's side of the family. She does have some interesting genetic disorders. She has a history of autoimmune disorders in various different forms, and I want to track that in my family, and I feel it as a responsibility as both a parent and as someone who has knowledge in this field to kind of move that or keep that in mind as I move forward with my family. But also, it's also just out of sort of the enthusiast curiosity. So, and in that sense, people often sort of point to people on stage going through their genomes and saying, look at them looking at their narcissistome. And yeah, for certain, there's going to be a lot of that too, because it's fun to kind of poke around and see what is, my, digital version of myself in some sense. But also, I think there will be things here I find that I think show the promise of being actionable. I wouldn't say that they're completely actionable, but have that promise. And as the field progresses, I am hopeful that exomes will have the power to be informative at the consumer level. Now, another thing to note is, the data that they're giving us is extremely raw. It is not a report that looks anything like their web interfaces for their known disease risk, etc. They're essentially giving us the level of data you would give a researcher who ordered an exome from a CLIA lab. And I think they do go through a large effort to make it to that quality where, one, they're using an ADX coverage, so we're getting, we're going to have high quality in the variants that at a clinical level grade, and some people have called a clinical exome 100x. You could quibble between these numbers, but this is essentially a clinical level grade exome. And two, they've gone through best practices in using tools and alignment strategies and really trying to give you something that would represent essentially the best any core lab could do. And I'll talk about that in a second too. But it is important to note that there's no interpretation, and that's kind of what I'll be getting into today. So finally, then, the question is, okay, they can do their best practice, but is that clinical grade? And are we looking at sort of a true representation of genomics here, or are there confounding issues? And the answer is, of course, there are confounding issues. This is still a largely research-driven field. The tools are generally built by researchers who are trying to always push the limits of what they can get out of their data, and the analysis is often based on large consortiums who are putting together great public data sets, but those are changing all the time. So it's very tricky to implement this at the same level of sort of clinical reproducibility and sensitivity to false positives, as well as the ability for false negatives. There's another point I wanted to make. Okay, well, there's three things here, and I want to cover them relatively briefly. So the first one is, in my data from 23andMe, I got an original VCF file, and then a couple months later they sent me an email saying, "Hey, we've been hard at work. We've updated our pipeline. We produced some more, a new analysis of your exome. Here's a new VCF file." And certainly there were more variants, and there's a little blog I wrote on Monday or over the weekend and published on Monday called "GATK is a Research Tool, Clinics Beware," because essentially I had the same data, the same sequence produced, and yet they ran it through two different versions of GATK, and you can see there was disparity between the variants called. That's all well and good if you look at the fact that most of these variants that were different were low quality, etc. What I actually found was there were these variants, and one of them showed up in my abbreviated report that they give you as a PDF with a big old red box around it saying you have a frameshift insertion in this gene that's rare, and I didn't have that in my first report. And that variant was one of these 8,000 variants I discovered were sort of phantom buggy variants. And in fact, I confirmed it with them that there was a bug in the version of GATK that they used in their latest release, and they updated GATK and the bug went away. So these phantom variants were essentially, because they're calling variants across multiple samples, my insertion that I saw in my report was definitely not in my exome. There was not a single read that supported it when I opened it up in GenomeBrowse, but it was in somebody's exome, and I essentially leaked the genotype leaked into my report, and it was essentially a false positive, but it was more than that, it was a bug. So that's just a cautionary note on the fact that these tools are still evolving at a pace that requires the type of clinical validation you really need anyway when going into a clinic, but it does have implications on what I'm doing today, which is still purely in the realm of a research level project. And finally, exomes are only that 1 to 3% of the genome that are protein coding. Is this going to be adequate if you're going to be talking about doing any type of clinical or even consumer level genomic work? Do we need to go to whole genomes where we're covering the full 3 point whatever billion bases? And I think the answer is, for now, especially at the clinical level and even at this level that I'm doing, I wouldn't know what to do with whole genomes. The annotations outside of the genes themselves are basically limited to, one, all those SNPs that I already have from 23andMe, plus all of their great interpretation involved with it, and two, all these regulatory regions that are a little less clear-cut. If I have a mutation that's in a transcription factor binding site, do I know exactly whether that mutation changes the expression of the gene that it's next to, or is it potential that the transcription factor still binds relatively okay? You need all this extra extra information like RNA-seq and your methylation information, and and it becomes very tissue specific. And essentially, genomics and biology is immensely complex. And so the sort of the nice thing about the exome is we have some reasonable way of doing a cause and effect between a mutation and a disorder, and that chain of reasoning requires a lot more advancement in our research as that on goes to do that same type of thing outside of the exome. But certainly the ENCODE project is promising in producing more of that data, and I really do hope to see whole genome sequencing play out to have a huge role in our both clinical and consumer level work in the future. And there's one more thing I wanted to point out with exome sequencing, and that is that at the end of the day, this is not a I guess there's a chance here that you miss things, and that's really the key point here, is that exome sequencing is really called exome target capture and sequencing. That target capture component is where you take your chopped up little pieces of DNA and you pull down the regions that fall within your exomes, and you'll see as I jump on over to GenomeBrowse in a sec, that gives you nice little curves around the exomes where the coverage sort of plops right down in the middle of that exon, which is great, but that capture process is subject to sort of the idiosyncrasies of that binding protocol. And as we all know from the genotyping era, those probes and those things can be very susceptible to batch effects. And in fact, EdgeBio put out a great little cheat sheet recently. And this is the critical thing here, is they're talking about essentially if you have a research grade exome, 30 to 50x, of the target region, i.e. the regions of the exomes that you hope to cover, that you want to be able to call variants in, you only get 70 to 75% of that on average at 20x, i.e. a level that's reasonable to actually call variants. And even with a clinical exome like the one I'm be dealing with, there's still a 5 or 15% chance that you're missing coverage on a region at sufficient depth to call variants. And I've seen this in my own data because I have a trio. I can do things like look at a variant and be like, man, I really want to know what that variant is in my wife or my son, and the data is just sort of not there. The distribution of reads just sort of drops off prematurely for whatever reason at that gene. And so this is a problem for clinics, I would imagine. And there's definitely ways in which they're trying to address this. But at the end of the day, if you're trying to produce a clinically certified test, you need to be able to say, if I have a variant within this region, I will find it with this level of certainty every time and get to the same result. And if you have a 5 or 15% chance of missing that target region, that's a problem. But there's also ways that you can deal with that, and I know for example, you can work on building a custom capture kit that only uses probes that are much higher chance of performing on target, etc. The other thing to note is this 5 or 15% is not the same percent for each person. It could be 5% for me and a different 5% for my wife, and that's what I was seeing as well. Okay, so then the next question is, let's go talk about what I want to actually do today, which is, look at my exome and how I want to filter it or analyze it, and that usually starts with a filtering process. Obviously, I could go at it a different way and say, can I annotate, the SNPs that are common in my exome, or can I look at more pharmacogenomic effects that are in my exomes? But again, 23andMe has already provided me, hundreds of thousands of SNPs, well annotated, and it's unlikely that just these regions of the coding regions are going to have more common SNPs. What they are going to have is lots of rare variants. And so what I'm going to use is an approach to actually look at those rare variants and see if I can find high impact variants that seem significant to me. And again, I'm going to be using for all this analysis, I've used Golden Helix's software, SNP Variation Suite. It does this in a very transparent way, and ultimately you could put all this in potentially a big package that you click a button and it does the exact same thing for every exome, and that's the level of reproducibility we hope to have with software. And certainly as a product manager at a genetic software company, it's very exciting to see all this coming together and have that possibility in sight. But it's good also to point out the limits of what you can do versus software versus what requires sort of that wetware of pulling your brain together and wrapping it around all the PubMed, OMIM, and Wikipedia data for a gene to really interpret it. And that's actually something that I do want to point out. There are experts who do this for a living. Genetic counselors spend their days trying to interpret genes and their impacts. And even with the public available data, I'm going to be limited in what I'm able to do. Not to mention the fact that a lot of the data that is useful for interpretation or interpreting the pathogenicity of variants, those are behind paid, private databases. Either they're private to a clinic, because some clinics do specialize in a disorder and have a lot of annotations, or they're in something like HGMD, which I do not have access to. So there's more public databases that we plan to see in the future, like ClinVar. And so I think some of this is going to continue to progress to more open access, but it's certainly worth pointing out that unless you're lucky enough to sit beside a genetic counselor on an airplane and talk to him about your genes, that's a hint that I actually happened to do that, you won't actually have, you're still going to have a struggling time doing the best you can with public data. So let's go ahead and filter my exome and do some analysis. So I start off with these 150,000 variants, and I'm aware of these phantom variants that I cataloged in my blog, so I'm definitely filtering those out. But I also want to just get down to a reasonable set of variants. Remember that report said using 20x as a cutoff, and we'll look at sort of the histogram here. I want to be very conservative. I don't want to lose information, but I do want to not look through a bunch of false positives. So I don't want to get down to the point where pretty much every variant called at 5x, for example, is complete crap. So we're going to look at some quality choices there, and then we're also going to filter down to rare. Again, what I'm going to find with exomes that I won't find in a genome-wide, genotyping test is rare stuff that's either unique to me, completely novel, nobody else has it but me, or is at a very low frequency in the population, and those are generally going to be enriched for highly functional variants. And finally, trying to ascertain which of those are functional, I'll use a functional classification tool or a variant classification tool in SNP and Variation Suite. Again, all these things I do in SVS. We have tutorials, etc. for doing them. I'm just going to essentially do this at a high level where I'm showing you tables of numbers and then get to the good stuff at the end. All right, so let's talk about that filtering strategy. So to start with, what I did is I imported my my VCF file. I have my 150,000 variants, and I looked at the read depth and genotype quality distribution. So there's 150,000 points that make up this histogram. This one actually I have it zoomed in at 100. It goes over to 250, at which point it looks like they did a hard filter and they didn't show me any place that has over 250 reads. Why would you have a place with over 250 reads? Well, it gets into the whole mess of like there are regions of the genome that are highly repetitive and short reads align to them very prolifically, etc., etc. But at the end of the day, these are all high quality up here, I would imagine. But you can see there's a lot of variants that are called enriched for these regions that have low coverage. And if I zoom in, this is a just that first 30 bases. And at some point, if I want to call a heterozygous variant with confidence, I need to pick a threshold where I can have a number of reads supporting it on each of the autosomal chromosomes, for example. So I'm going to pick a threshold of 10 here. And also I'm going to pick a threshold. Well, I want to know what I really should do is figure out what, does this threshold equate to this one? I mean, if I filter out read depth of 10, does that remove everything down here? And let's go look actually in GenomeBrowse to answer that question. So I'm switching on over to GenomeBrowse, and this is our free genomic visualization tool. Remember all the analysis I did in SVS, but this is a great way to validate any individual variants and sort of your analysis in general. And I'm going to show you a couple things in here that we just released in our 1.06 version that came out last night. Oh yeah, I was definitely here last night. And so we're going to actually just spend some time going through a couple variants back and forth. But to start with, let's look at, let's see, I have a little bit of documentation I've been working on here. A variant that has, surprisingly, a genotype quality that's quite low in that sort of less than 10 range, 3.8, and has a read depth of quite high, 66, and see what's going on with a variant that follows that pattern to see if we want to filter those out. So I can click on this. This is some documentation I wrote for myself just as I was doing this webinar, but as I click on it, I essentially jump to that variant in my exome. So here it is. It's right centered in the screen. I'm going to kind of highlight it so it's easy to see. It's good to point out a couple things about GenomeBrowse. One, I'm scrolling with my mouse wheel here, so it looks like magic, and it is really, if you think about it. But what I'm essentially doing is just looking at the genomic context around this gene, and the fact that I've got some annotation tracks here. I have the OMIM database up, and I have this track, which I'll talk about a little bit later. But if I get back into this individual variant and just fly right in, you can see the coverage is centered on the exon as you expect. But the variant itself seems to be, essentially have very few reads. If I click on it, there's 6 reads that support the mismatch and 74 that support the match. It's also suspect is if I can click on this to show various different ways of stacking this. I'm going to stack above just to get, this is the coverage information, and this is the sequence reads themselves. It's also suspect all the sort of mutations going around in here, and in fact these gray reads mean that those reads are ones that have mapped to multiple parts of the genome. Essentially there's paralogs of the genome that this read maps here as well as another place. One of these features we added just in our last release was the ability to filter out those. And now that I've removed those, you can see my depth of coverage shows that I have 40 reads that aren't multi-mapped, and still only 6 of them are mismatched, and my depth is dropped by half. And so it's quite obvious that this is a problematic SNP, and it's a good thing that the genotype quality is low, and I definitely want to filter out variants with this problem. If I notice also up here I have a track called superdupes. It shows me that this region is, if I click on any of these things, I can see that this has actually 95% similarity to another region of chromosome 9, and not only that, it has 95% similarity to a region of chromosome 20. So this is a region of the genome that has a high level of duplication, and we're going to actually use this track later as a filtering technique as well. But fair to say that I can drop variants that follow this pattern, and so I'm actually going to choose a quality threshold of 10 here and drop those guys, and that did drop my variants down from 151 to about 80,000. So the next thing I want to do is talk about how that variant classification and the frequency classification brings us down to a more reasonable number. That frequency classification is using 2 catalogs. I'm using both 1,000 genomes in its latest release, so that's 1,000 individuals, as well as, and those are at a whole genome level, as well as the NHLBI 6500 exomes, which is 6,500 individuals with extreme phenotypes like I mentioned earlier, and their frequencies are cataloged across the exons, or they have an exome capture for those 6,500 individuals, so those frequencies are going to be quite accurate, and we'll be seeing at a different point that I can look at more details like their genotypes as well. So just knocking down to rare, I have quite a few less, but also when I classify my variants, you'll see that quite a few of my remaining variants are intronic, i.e., like I was showing you that one in GenomeBrowse, it sits right outside of the exon, or in that intronic region between exons. And some are still upstream or downstream from the genes, some are in the UTRs and in genes that are not coding. And then the ones that are in the coding region, i.e., they are right inside those exons or in that splicing region between exons, those 2 base pairs intronic up at the acceptor and donor sites. So of those, there's quite a few that are synonymous, that are single base changes that essentially do not change the protein sequence. There are some insertions and deletions that do not, that insert a single or multiple amino acids but do not shift the entire protein sequence encoding and infer a complete damage to the remaining rest of the coding sequence. And there's some that are unknown because the annotations do not actually represent full protein coding genes. So what is left then is these 3 highly categorized variants groups, which are splicing variants, frameshift insertions and deletions, and stop gains and losses. And these ones we expect to have a high impact on those proteins. Yet at the same time, there's a lot of them. There's still 200 of them. And then even more are all these missense mutations that are non-synonymous, 1,500 of them. In parentheses, I have the number that are completely novel, which are not in dbSNP, etc. So there's still quite a few number of variants to plow through here, and I need some more filtering strategies to break that down. So to start with, what I'm going to actually do is just like that track I was showing you, we're going to look at removing variants that are in those regions of chromosomal duplication. The idea here is, one, it's ripe with false positives, and two, those genes that are completely duplicated with high similarity most likely have paralogs that can sustain a mutation or a loss of function in one gene. The other thing is I'm going to look at genes that are in OMIM, I'll show you that in a second, and ones that are predicted as a more restrictive filter, ones that are known to have a recessive disease associated with it or predicted to be able to hold a recessive disease variant and have it be pathogenic. And then for our non-synonymous variants, we can look at some functional annotation information, which I'll show you too as well. But what I want to do is kind of talk about those tracks and those filtering strategies in the context of GenomeBrowse. And to do that, we're going to start with actually just looking at another variant here that is in a super duper region. And this was a rare splicing variant. So if I click on this guy right here, and I'm going to see there's no other variants that are present in the control databases, so I'm going to hide those real quick and just focus on the fact that it is within those first two bases of this intron. So you can see if I zoom out a little bit, the variant I'm actually looking at is this 4-base deletion, so it's a frame shift deletion. Notice I have my filtering on, and half of the reads here again are multiply aligned, and we're in a region of segmental duplication. Also, more interestingly, even though this is a frame shift insertion, or sorry, a splice variant, you can see that if it is true, and I think it definitely exists on one of my chromosomes, if we were to remove these 4 bases, the consensus sequence would still be GTA, just like it was before. So it essentially does not actually impact the splice site, even if it exists. But as we zoom around here, you can see these big chromosomal duplications and the issues around them, and we're essentially going to remove variants that are in the genes that fall in those. And they're not all over the place, right? So they're here and here, and we're in this, notice we're right in this region near the centromere of chromosome 2, which is not that surprising that these are regions that have high repeatability. And the other thing we were saying is we're going to use OMIM genes, and notice that there are a lot of OMIM genes. So we're going to keep things in here. There are definitely genes that are outside that, such as this guy here, but these are going to be generally genes that have no known biological implications for having there's essentially no publications implicating them in any biological process based on their mutations. And then that last filter I said was based on this likely recessive track. So I have these 2 tracks. One of them is called DB NSFP gene annotations with NTRES gene coordinates. And essentially, if I click on this, for every gene, it gives me a little description about its pathway, etc., and some prediction about its likelihood to play a recessive pathogenic role. And this one has a 0.1%, very low, or essentially a 10% chance, very low percent chance. And so I have a track here with known, notice the triangle ones are known recessive disease genes, and they have descriptions about the disease and what they're doing here, as well as the fact that that they have a little bit higher percentage of being predicted as recessive, and some that are not known but still have an extremely high percentage prediction of being recessive. And if you were to look at the public mutations that are present around here, if I pull up, for example, NHLBI, these guys, what you generally see is these mutations don't occur in a homozygous state because essentially this gene is likely to be extremely important to stay conserved. And my own genome, if I look around here, it looks like I have no variants, and I would have to pop into each and one of these exons to see what's going on, but I seem to be relatively conserved. So we're going to use this track up here of known likelihood variants as well to help with the filtering. So let's go see what that looks like. So again, all this analysis that I'm doing is in SVS, even though I'm showing you the results in GenomeBrowse, and we're going to step through some of the results in a little more detail. But using those filters, I can knock down the number of variants by about half that are outside of dupes, and then again a little bit more based on those that are in OMIM genes. And it seems like, going to that recessive list is very aggressive, and maybe we don't want to look at that as the final result. I definitely am interested in variants first that are homozygous in myself, right? So if I have a heterozygous mutation in a gene, it's very possible that even if one of that mutation completely knocks out the gene on one of my chromosomes, biologically that is acceptable and it's essentially haplosufficient. With one copy of the gene, I can still perform the regular biological functions necessary for that gene, or quantitatively reduced but still not symptomatic. At the other hand, if I have a homozygous mutation where I have both copies of the gene contains the mutation, there's a good chance that if that gene was critical, then that gene is no longer able to play its role, and that could lead to the pathogenicity of disorders, etc. And so those are ones I want to look at first. So I have 16 mutations only that are homozygous in OMIM genes at this point. Some of those are these frameshift insertions, deletions, and splicing variants, and we're going to actually just go through a lot of these one by one. And then for my non-synonymous variants, notice I broke it down here by this sort of voting score. We use that DBNSFP annotations for functional predictions. That gives me 3 different tools that say whether they think that mutation is going to be damaging to the protein. And if all 3 of them agree that it's going to be damaging, I put 3 out of 3. If 2 out of 3 or 1 out of 3, I rank it like that. If they all think it's going to be tolerated or they weren't able to run, I have some of those. So essentially, of my homozygous variants, 4 of them are sort of expected to be tolerated by all of them. But again, these are tools that are probabilistic in nature and are not, or have been shown not to be much more than 80 to 85% accurate. So I definitely don't want to dismiss these, but it is information to use to help rank things. And finally, of my recessive genes, I have very few that are homozygous and in these recessive areas, and we'll look at those for sure. So at this point, we're really actually going to spend some time plowing through some variants, and it's already I don't have a whole lot of time here, but I am probably going to just let this run over the top of the hour because this is kind of the fun, meaty part of it, and we'll take questions afterwards. To start with, I wanted to show you kind of how I got to this point in SVS. Not like a blow-by-blow, but I was able to do this filtering, all these steps, essentially. As I pull up one of these things, it shows I'm starting with my outside dupes, and then outside dupes and outside of OMIM or in OMIM, then my known likely recessive, then the ones I'm homozygous for, etc. All that annotation kind of builds up step by step, and I'm able to pull up a spreadsheet like this one. I sort of highlighted or de-highlighted this one we're going to talk about in a little bit, where I have these 16 homozygous variants and all the annotations for them, including their HGVS nomenclature. This is how you commonly describe variants in research papers as well as in common databases, and their allele frequencies where they're not completely unique to myself, and only a handful of these are present, as well as their genotype counts, which is quite important, especially when looking at homozygous variants to see if other people contain them in a homozygous state. So let's go ahead then and pull up GenomeBrowse and go through some of these. So I'm going to pull over my sort of notes that I've taken. I want to keep that popped out actually. And I'm going to scroll on down to some of these homozygous variants. So this is a frameshift insertion, and I'm going to click on this and see what's going on here. And drag this out of the way so it's not distracting. And I'm going to scroll on down to see, I'm going to hide super dupes because we've already filtered against that. I'm going to hide OMIM because we know we're going to be in OMIM genes, and bring up 1000 genomes in dbSNP. And then look at what I've got going on in myself here. So this is definitely a homozygous mutation, and it looks like it's insertion of a C199 read supporting it. Looks very clean. I don't distrust the call at all. There's a little heterozygous deletion a little bit away, and it's definitely here, and it's in dbSNP, but we filtered against these 2 tracks, and it's not in here, so it didn't fall out of our filters. Now I'm going to do something like pull up my wife's genome and just see what she's got. And she's actually got the same thing. She's got the homozygous insertion, and it's in dbSNP. And because we're unrelated, I basically can consider this a common benign variant if we're both homozygous for it. So I'm going to hide her and dismiss that one. Scrolling on down, I'm going to actually just jump us to another site. And this is another site, and again, give us a little context here, that we're right in the middle of a exon, so this is a potential frame shift insertion, and it went through all of our filters, and it's this little guy here with 32 bases supporting it that fell through our filters. You can see there's no insertion here, but there is one, and I'm also heterozygous for this guy, that is present and got filtered out. And if I click on it, you can see that that's actually quite common. So the question is, are these on the same haplotype or the different haplotype? In other words, am I homozygous? Notice it called me homozygous, but this is clearly a heterozygous. So one, it's a bad genotype call to start with. But two, if I zoom in on these actual reads, you can see this pattern that the heterozygous call for this site is on clearly supported on forward and reverse strand reads, but on a different chromosome, on a different read entirely than the one here. So I essentially have just another mutation on my other chromosome, but because there is a common mutation, this frame shift insertion must not be that interesting if half of the people have it. So it's essentially, this is classified as benign by inference. Something you really need to do with GenomeBrowse. You can't necessarily bioinformatically pick this out very easily. Certainly I hope to make a lot of these false positives fall by the wayside as we improve software in general, but this is a tricky one. You could definitely see it's sort of benign by inference here. Obviously the population catalogs will improve and these variant calls will be present more likely in the future. So jumping on over to this guy, so I'm definitely homozygous for this insertion. It's right here on the beginning of an exon, so it looks a little bit scary. It's present in dbSNP, and it's also present in 1,000 genomes. So let's see how common it is. Wow, it is so common it doesn't occur in the reference state at all. So it's 100% in the reference genome. And so the question is, why then is it still in here? Because I thought I was filtering by alleles less than 1%. And the reason is because I was filtering at sort of a variant level, not at a genotype level. And because I'm homozygous, I'm actually homozygous for the reference, right? The reference, everybody has the C. If there was a non-reference, if I was heterozygous or if I was, no insertion, that would actually be rare, because the common thing is for everyone to have that C. So we kept it in the filter because as a mutation of an insertion of a C at a heterozygous state, that is actually a rare event. In my case, it's completely benign because it is the real reference. And this is not uncommon, and something you have to keep in mind is that the reference genome does have plenty of sites in which the reference is the minor allele. Okay, so here's another missense mutation. This is one of my non-synonymous homozygous missense mutations inside of a gene. What's going on here? Well, the context is immediately a little suspect because you can see from the reads and the kind of the mess of the mismatches going on that something's going on. It looks like for the reads that don't support the G, there is this bad alignment going on where there's an extra mismatch and an insertion of a GA, etc. We're not in super dupes because we filtered that out, but it does seem to be problematic. And then if I pull up dbSNP for this, this isn't dbSNP, right? If I pull up dbSNP for this, and I can just throw that onto the screen from my other screen, sorry for the sort of jargon thing, this little icon actually means that dbSNP views this as a suspect genotype. And if, in fact, if you were to search for the actual RSID in dbSNP, it's a little more clear. They have a big suspect. So basically they have not a lot of confidence that that's a high quality genotype, and I think I can safely ignore it as well. Okay, so now I'm going to look at a another homozygous variant. This is supposed to be a deletion of a T. Well, where's the deletion? I just see a nice C mismatch here, and that seems to be fairly common. A lot of people have a C here. It's actually 99% of the time. It seems to be almost the reference. But if I click on it, I can see down here in my table that I do have one deletion. In fact, if I want to, you can always unlock these graphs and really zoom in using a little trick here. And you can see there actually is that deletion here. I'm going to lock it back up and go hunt for it. So if I can just sort of find it, oh yeah, there it is. So I do have a deletion, but but why the heck was this called with high confidence? And in fact, if you look at my project notes, that table shows this deletion was called with 99% confidence. I'm pretty confident that this is just another bug in GATK. The deletion was overcalled with confidence. It might have been that there was a sample that had a high confident call, and that information sort of got propagated into my data. But that's just plain out wrong. That's not a genotype, and that's just a bug. So finally, let's look at another insertion. Now, I just centered my view on this insertion, but I don't see an insertion. I'm supposed to be finding an insertion of a CC, and it looks like there's an insertion of a GG, but I don't see an insertion of, oh no, there is supposed to be an insertion of a GG. So again, this is another one of those cases where the genotype is there, it's at one basis, but the variant was called with way too much confidence and should have been filtered out as a complete false positive, because this is just a buggy variant in GATK. Okay, so those are a lot of false positive I just filtered through. Now I'm going to actually show you some real interesting, things I can't dismiss as false positives, and now I have to try to interpret them. So this guy, I just jumped to it. Notice I'm right here. I'm going to give us a lot of context and zoom way out using our big old zoomer here. I'm right in the middle of this section of my X chromosome. Given that I'm a male, I only have my one copy of my X chromosome, and what I expect to find here, if I am homozygous, is that I'm actually hemizygous, right? So I have only one potential allele. I'm right in the middle of this gene. It's a big honkin' gene, and it does have annotations for my my DBNSFG annotations. If I click on it, I can get some information about it. It doesn't have any known disease phenotype information. It also doesn't have a really high recessive probability score, so maybe I'm not too concerned about it. But at a variant level, it's very solid. I have 60, this is truth to me. Like, I have 60 G's representing the insertion, the mismatch. It's non-synonymous. It's actually completely novel. It's not in any other database. It's on the forward and the reverse strand. I can split by strand to make that more obvious. And it's just a real clean mismatch. Now, how do I want to interpret this? Well, as I mentioned, I actually was lucky enough to be looking at this mutation while sitting on a plane on the way back from ASHG, and the person sitting there happened to be a genetic counselor, and she was asking me about what I thought about my mutation. I was like, well, don't ask me. What do you think about it? And so when she got off the plane, she went ahead and looked it up, this gene at least, and given that there's not a lot of disease pathogenicity known, she went ahead and looked up to PubMed and found that there were 6 citations relating mutations to this gene to sensitivity or other different toxicity reactions to some common chemotherapies. So basically, she suggests that it'd be in your best interest not to get cancer, which of course I'd be like, yeah, I'm on board with that. Let's not get cancer. But at the same time, if I do ever get cancer, I would be monitoring this gene very closely and monitoring the chemotherapies used and the publications that continue to come out for it, because there's a chance, even though those prediction tools did not predict it as being likely disease causing, that could be a real trait that I would want to be aware of. So finally, I'm going to look at this non-synonymous variant in OTC. This is essentially one of the other non-false positive that's worth looking at, and I clicked on it, so we just jumped to it, but it is actually predicted as tolerated by all 3 tools. Again, it does have a really high conservation score, and if I look at this variant in dbSNP, this whole gene, in fact, is highly implicated in a disorder. And if I look at this individual variant, it is listed as a pathogenic variant, and you can actually look at this VAR view here in which they show you all the variants that are present in this gene, and the variant I have here is highlighted. It's known pathogenic. There's 2 papers in PubMed about it. I tried to read these papers. They didn't list my variants, so I'm not quite sure how those get associated. But essentially, and let's go back to GenomeBrowse to take a look at this thing, it's definitely a high quality variant. 33 read depth. It's not present in 2 of the catalogs here. It is present in dbSNP. That's what I was just looking at. The context is very clean. It's essentially the only variant in this gene that I have that are in the exons. I can scroll around to the other exons, and there's no other exons which I have coding variants for. And so this one right here, for example, is right outside the intron. So back to my variant that I was looking at. I'm homozygous for it. And if I click on my DBNSFP, it is predicted as likely pathogenic if inherited recessively, and it also has some pretty good description about its actual phenotype. Basically, if inherited, or if the mutation is in its severe form where it impacts the gene directly, you essentially have a fatal disorder at birth in which more than half of newborns die within the first 72 hours, and then another half in the first month, and another half by the age of 5. But at the same time, it's also common to have late onset version of this. And it essentially is a metabolism issue where you aren't able to process some heavy proteins, and you get an extra buildup of some ammonia and other very strong clinical reactions to that. And so this is actually something that I'm going to take very seriously and try to follow up on. At the end of the day, I'm still young, and so I could keep an eye out for a late onset condition here. I could also look at the fact that this is inherited on my X chromosome, so my mom has to have this in either a heterozygous or homozygous state. She's actually been one never to be a big meat eater. She's been a vegetarian most of her life. She eats fish every once in a while, and more recently has been even eating less meat. So I might actually follow up on this and see if there's worth doing a clinical test and having her or myself see a physician, because there's some known dietary and other types of supplemental things you can do to help sort of ameliorate this deficiency. So this is kind of a real interesting hit for me, and I'm definitely going to keep an eye out for this. But if I were to go back to the slide form to wrap this up, the 14 homozygous variants I had were mostly false positives. I went through almost all of these. They're either in my wife, so they are not rare at all. They're shown to be common by some other form. They're bad variant calls. And then there's these couple of variants of unknown significance. This one actually has some allele frequency that's relatively common. This one, I didn't show it, but it was in 5 other males at a homozygous state as well at the NHLBI group. So it's in 5 other adult males, but again, those males have extreme phenotypes as well, so not necessarily dismissed. That's that pathogenic variant that I looked at OTC that I'm going to keep an eye on, and here's that variant of unknown significance that my co-flight genetic counselor helped me out with. So this is all kind of well and good, but at the end of the day, it's still important to note that the technology, the tools, and everything is still progressing here at a research phase, and I'm going to continue to essentially reanalyze my exome. And again, I'm not going to be sharing any of this information when I start looking at my wife and my son, but I think it will be interesting to look at the ability to look at a complex trait such as an autoimmune disorder and see if that can be tracked at an individual level, because at the end of the day, the research has all been done at a population level. We can say which genes, which hundreds of genes are implicated by autoimmune disorder, but it's very rare to have a single or even a set of mutations be known to be linked. So, this is the kind of the success and the limitations of this type of experiment, or this type of assay, is that we can look at rare variants, but we can also get to a lot of variants that are essentially leave the door open for interpretation. So, and that's really kind of what I hope to cover today, and we did go a little bit over time, but I hope there's still interest in taking some questions, and I can see there's quite a few people still on the line, and let's go ahead and plow through some questions now. So the next question is, how long are the samples kept? 23andMe basically determines one predisposition for developmental of certain diseases based on their DNA. So that's a good question because I don't know if they're keeping my saliva per se on file. They ran the genotype chips, so they have that DNA and the genomic information there. I know they reprocess that as their algorithms improve, and similarly they have my raw and processed exomes. So they are probably going to keep the digital versions of that, but I don't know if they're keeping a saliva form of it. And I think that's actually true that they don't, because I know 23andMe actually participates in the research community, and so they allow, for example, a researcher to say, I have this extremely rare disorder that I'm trying to find, recruits to be part of my study, and 23andMe send out a notice to people who are likely to have that disorder, and they can volunteer to participate, but you have to send in some more saliva because you're going to end up getting a new run of genetic testing done. So one of the questions is, does this data only go to the consumer, or do they keep it on file? Clearly they do keep it on file, otherwise they wouldn't be able to send me an email saying, hey, I got another version of your data processed. And that makes sense because they're also at the same time, and I think for the benefit of all of us, trying to determine, can this be coalesced into a platform where this interpretation that I've showed you, which is very meaty, can be displayed to a user in a meaningful way, sort of in an automated and meaningful way through a web interface. And that's going to be essentially a big open research project that they have to continue to work at. Another point of that mean coverage is a terrible metric. What percentage of the exomes is covered over 20x? That was a very good point, and I think that was what I was mentioning in the exome cheat sheet from EdgeBio, is that with an 80x I could probably expect to see 80 to 85%. I should actually run the metrics on my own exomes to see what percentage of the exomes was covered over 20x as well. That's a good point. I'll keep that in mind if I'm going to do a further blog post to compare that to sort of what is expected from an exome capture. For the exome, the question is, what exome capture kits did they use? Shoot, I wonder if I have this right off the top of my head. I believe they're using a Nimblegen exome capture kit like V2, and they're using BWA and GTK for their pipeline. They've also, it's on my blog and it's on other people's blogs too, so it wouldn't be hard to get that too with a Google search. So is this can you access this data, or can you access the same platform yourself? Yeah, I mean, researchers are using, like I said, 23andMe is essentially trying to follow a very best practice approach. They're using an exome capture kit and a alignment and variant calling strategy, very similar to pretty much what any lab setting up a exome pipeline might do. So question is, what does it mean to have a common SNP or a novel SNP? Well, there's actually 3 different classifications I'm going for. One is common, which is it's going to be in dbSNP for sure. It's also got an allele frequency of greater than 1%. Of course, that matters which population you choose. All the filters I did was based on European population frequencies only, so I'm not filtering against, say, the African American or Yoruban or whatever. There's actually multiple population groups in 1,000 genomes, as well as multiple population groups in NHLBI. And so I filtered against an ethnically matched population group of 1%. Novel means it's not in dbSNP, but to me it also means there's some things that are not in dbSNP that are in NHLBI. Novel is there's nobody who has it, right, that I can find. And so as population catalogs improve, we expect to see fewer and fewer of those, but I think I saw a metric once that for every exome you sequence, you're going to find on the order of 1,000 novel variants for someone who's unrelated to the previously sequenced people. And so there's going to be, this catalog is going to improve, but it's going to keep on going essentially forever because of the wide diversity of mutations that can be privately held. What's the probability of missing an already known rare pathogenic variant? So this is where I would love to have access to something like HGMD, which is a commercial database that catalogs all the known pathogenic variants. But in the future, ClinVar will have a lot of that data publicly available for enthusiasts and researchers and pretty much anybody as well. And that is often data that comes from the benefit of labs sharing that. So if you're a lab and you go through the hard work of nailing down the pathogenicity status of a variant of unknown significance, and you publish not only your work but your thought process and the evidence that you have, and then that's all well and good. That seems benevolent, but then the sort of benefit to the lab is then somebody a world away can find that exact same variant, and find that exact same variant with the same phenotype and confirm that pathogenicity status that was relatively putative until you have a second causal link, because that's the intention of these things to be highly penetrant is the same variant should produce the same phenotype. So when you find a second family, and everyone benefits from the sharing of that information. So there's definitely at this point with the analysis I did plenty of chances for me to be missing stuff. Again, that's why I plan to keep my data on file and keep coming back to it and saying, I got new databases, I have new ClinVars out, let's annotate this variant, let's annotate my variants against that. 100x coverage will be better than 30x, you betcha, but at the same time 200x coverage might not be that much better than 100x. I hope I made that clear. Are the results interpreted to participants in the detailed way you did to us today? Yeah, that's a great question. So basically, do other people in the 23andMe Exome Pilot have the same type of analysis results interpreted? And the answer is no, definitely not. They're all on their own because, like I said, they've just been giving this a sort of raw dump of data. At the same time, the tools I'm using are commercially available. Golden Helix SVS as an analysis tool is, our bread and butter, and GenomeBrowse is a freely available tool that we give to the community, so you can look at variants and all those annotations I showed you with not much more work than just downloading, registering, and popping up your BAM file. Of course, you need to know where to look, because if you were to pull that sucker up and just start at the whole genome level, you could spend a long time getting nowhere. So you definitely need that type of analysis tool to do this type of work. And I think what 23andMe is looking at doing is seeing how much they can automate that, and as well as we're continuing to work with the clinicians and researchers out there to give them tools that make this much more automated and robust and something you can deploy with minimal effort. Does 23andMe supply you with a breakdown of your racial background? Oh, that's a great point. I didn't touch on that because I was just focusing on the traits, but they do have a great ancestry tool. And so I was able to look at sort of my paternal and maternal ancestry, given I have both Y and X. My wife, though, can only look at her maternal ancestry. And it's actually really quite informative. They use some very robust tools like principal component analysis, and they have a very large population of individuals involved in 23andMe. So they're really in one of the best places of anyone I know to give you a very detailed ancestral background of both your haplogroup as well as a lot of sub-details about sort of your ancestry and information. So I found that very informative. Found some interesting things about kind of my father's side of the family that sort of matched up to rumor but went on even beyond that. So I mentioned, how do you get GenomeBrowse? Again, it's free. Please go get it. We're having a little contest to see how many downloads we can get by the end of the year, and you'll make me very happy if everybody on this webcast downloads it right now, and then I win the contest. This is a good question. What's the clinical application and area of curing certain genetic diseases using exome sequencing? And that, okay, so there's two major points here. One is there value in getting to a diagnosis where there's no known cure? And I think the answer is yes, and there's been studies that show just the level of burden on a parent to not have a diagnosis, because you can have a mutation that's completely de novo. It has nothing to do, there's really nothing the parent should feel guilty about, but even if it is a recessive mutation, to have a diagnosis is a win in of itself. And the second question is, diagnoses do lead to treatments. Definitely the success rate there is much lower than just getting to a diagnosis, but in the case, for example, of that exome of the son of my friend at the University of Utah who's continuing on with his diagnosis, he has a completely novel disorder. They understand the pathogenicity. They understand that there's a protein that's misfolding, building up in the blood, leads to the developmental delay and lots of metabolism allelic issues of their son, and they need to figure out if they can really solve it. And now that they have this information, they're able to, and the profile of that he has by being a sort of internet celebrity for publishing, or talking about this, he has had help from people who are willing to put him into clinical trials, proteomic researchers that are actually specialized in some of those proteins, and so the chance of finding a cure is certainly much more than if he didn't have a diagnosis and he was at a dead end. So the hope is there, and in a lot of cases, and there's case of whole genome sequencing success stories, the diagnosis did lead to a very clean and individualized treatment that essentially either totally made the disorder asymptomatic or managed the care in a much improved way. Okay, so this is a nice controversial question that is probably worth addressing as well. People are asking about, the age of my son. Did he consider his autonomy before embarking on this project? And very fair point. At the same time, minors are still under the provision of their there are still dependents, and I'm responsible for him. And at the same time, also, this is still consumer level information that does not impact his ability to, say, get health insurance or get a job. So it's a very important distinction to know that, the genetic test that I did does include my son, but it is not a genetic test. It is not a clinical test. It's information that I have and that I can use individually, and I think I can actually use it to help in the long run much more than the risk of not knowing something or knowing something that is potentially life-changing. So this is a huge ethical question, right? Can you make is the risk of finding something out that is so life-altering that it ruins your life that it'd be better not to know? And I just don't really buy that. I think it's always better to know, and it's better to take action, and that's the choice of parents in all sorts of ways with their children. And genomics is a immensely familial, private information, so I don't see how that's really all that different. But again, this is one of those conversations that can easily go offline, and I look forward to those debates in the future. So Golson Lyon, who I mentioned, is the author of the publication and the physician involved with finding the rare genetic disorder in Ogden syndrome that I showed was reproducible a couple months ago, mentioned that there's a post by Peter Kovitz on my blog and saying that it's neither a research nor healthcare, that this is a consumer level product. And ultimately we do have to have a debate as a society about what the right level of protection there is for consumers. We do have things like CLIA certification, which means that consumers should not be getting results from labs that are uninformative, and we should be protecting them from essentially snake oilmanship and other types of risks of consumers being exploited. So I think that's a fair point, that we need to consider this in the context of the chances for exploitation here. And I think 23andMe has done a very stand-up job in doing what they can to both make it very obvious what I'm getting myself into. I am getting raw data. To me, that's very satisfactory because I want to plow through it. And at this point, I don't think, just like you couldn't throw away everything but the final results of a test in a research setting, there's not really, the test hasn't emerged as such a slam dunk thing that I would be satisfied with just a high level report on exomes. At the same time, the genome-wide stuff is there. I don't feel a large need to pick through my own raw genotypes, even though I have them available, to see if I can find different GWAS studies or other traits or pharmacogenomic effects. I think they've gotten that nailed. So to me, this is the right level of access, but it's obviously still an ongoing research question of how this can be translated into something equivalent to what we're doing for genome-wide association tests. So a question is, with 23andMe, how do you handle research studies? And the answer is they are completely opt-in, for that. They allow you to participate in research studies, but they essentially act as a very large group of people with known genotypes that can participate. And in fact, you don't even get an email offering to participate unless you said, I'm interested in participating in research studies. And the fact of the matter is, it is extremely difficult to get a cohort of individuals together that represent a rare disorder and do any sort of significant size of that. So this is really almost a service that is unmatched in being able to have a ready participatory group of people that would like to see the research move forward and would be willing to put themselves into a research context. That said, that's one line I would not cross. I wouldn't put my son into a research context any more than I would want his genetic information in an actual clinical test on file. So that I think is, and it has been shown to most people to be viewed as a requiring consent of the individual more than anything. Okay, so the next question is, is this interpretable to the consumer? And I think I answered that fairly well, that this is still relatively research-oriented investigation, and the interpretation, the level that would be required to sort of coalesce this information into consumable for a consumer is probably far away. At the same time, I don't think the idea that we should sort of paternalistically hold on to all this until it's ready for Joe's six-pack is the right approach either, because for example, there are those enthusiasts that are driving their own care because they're really the only one with sort of the attention and wherewithal to put it all together and move it forward. And they may certainly spend the time to be able to learn to do the things that I did here and make sense of it. So we shouldn't also essentially babysit what a consumer should be able to do. But at this point, I definitely don't think it would even be popular to have this level of analysis available to consumers. So another question is, what about a thumbnail sketch of carrier status, traits, drug response, and disease risk results? So I don't feel like it's that useful for me to share my information on that level, but at the same time, you can go and get your own 23andMe results for $300 or whatever, and it's a lot of fun. The information I find there is quite useful, drug response, traits, carrier status, etc. But the point of this wasn't to necessarily explore my narcissism to the level of just dumping it all onto the screen, but more demonstrate the capabilities of software and the interpretation using public annotations to get to some meaningful results. Yeah, so a question about what I was using for that protein coding variant functional prediction. I was using, let me show you guys what I was using. So when I pull up my annotated variants here, this guy is predicted as tolerated, benign, and polymorphism, and this is the one in that OTC gene. So actually all three predict this as relatively benign. By all three I mean SIFT, polyphen2, and mutation tester. The analysis is actually, it's not that SVS does these predictions. These are all independent research tools. We're actually using a database called DBNSFP that collates all this data for every non-synonymous change in the human genome. So essentially any non-synonymous change you have, we can pull in what the predictions are. Notice they're not necessarily going to cover every non-synonymous change. This one doesn't have a prediction. It's sort of -1, meaning I don't have a prediction. Same time, we also have the sort of a couple conservation scores. So yeah, we're using polyphen and SIFT, which was a question about what we're using here. Oh, good question about the advantage of exome sequencing over RNA sequencing. RNA sequencing is important to keep in mind, and to be honest, it'd be really cool to have some of my own RNA sequencing data to help interpret some of this. But at the end of the day, RNA sequencing is almost a group level activity. You don't ever look at your data and know absolutely what's going on. You have to compare the frequency of expression in your data versus somebody else's. So for example, if I go to my add dialog here, do I have some RNA-seq data copied? No, I have some in my examples. So we have some cloud-based RNA-seq data that you can stream directly into GenomeBrowse. Forgive me if this sort of plows through my network connection and slows down, but I can add RNA-seq data and stream it. You get a feel for what it is. But the point of RNA-seq, well, there's no expression at this gene. Figures, huh? Here we go. Here's a gene that's got some expression. So the point of RNA-seq is that you're actually looking at the mature RNAs where those are going to actually, for those splice events, right, are going to just span from one exon to another. So you can see as I zoom in here, there's just a very small level of expression, but this exon is split by this read, which spans it, and you can kind of see for every exon, and there's potentially alternative splicing, etc. At a quantification level, if you expect to see high levels of quantification in a gene versus low levels, or really what you're looking for is a differential levels of expression, that can lead to sort of a biological crumb trail to getting to a diagnosis. If I had expression at this gene or didn't versus a sort of control, and I'd really have to do it at a large enough level to make that statistically significant, it could help me inform whether my mutation helped to or sort of disrupts the expression. Because disrupt expression means disrupt levels of proteins, which means disrupt, sort of effect on that pathway. Good question. Whether tools in SVS used to annotate splicing variants to determine if they are damaging. Yeah, so there's a whole different world in which people try to predict the level of potential impact a splicing variant can have. And we don't really have a lot of that data curated, but I am planning on adding from this track here. So we have this DBNSFP genes, but there's that prediction track that we were just using to do our functional predictions. And this is really dense, so don't be too surprised by it. But essentially, this lists every single non-synonymous change and their predictions as tolerated benign. They've updated to include a couple of those splicing tools, and we plan to update as well. So what we'll be able to do is, if there was a splice variant I had right at the end of this intron, those tools that do predict their functional impact, I'm going to just delete these guys, can be used to help rank those splice sites. In my case, I did not have that many splice sites to really plow through, so it wasn't that much of a problem. And splice sites are generally quite, rare and high impact by default. Yeah, so the question is, can my exome sequencing explain function or non-coding area illustrated by ENCODE? Again, you can see the pileups here. The coverage is going to be centered on the exons, and where I do have a little bit of overlap into UTRs, etc. Outside of the genes, there's limited annotations that have a real causal effect. So if I have a mutation in a transcription factor binding site, that might help regulate the gene, but without RNA-seq data, that's still a bit of a guess. I think ENCODE's this great experiment in sort of throwing a lot of different experiments together to try to collect data, but it's still an immensely difficult problem to correlate essentially, ChIP-seq, RNA-seq, DNA variants, and all that. So it's still, we've got a long ways to go before we're annotating variants outside of the exomes as easily as I've been doing this. All right, let's wrap it up from here. I hope I did answer many of the questions. It looks like there's a few lagging ones I didn't get to, but feel free to follow up with me on Twitter or email. I love to keep the conversation going. It's been a blast preparing for this webinar, and appreciate everyone sticking around even to this delayed or later question and answer session. So thank you very much, and I appreciate all the comments saying that it was a great presentation and they got something out of it, and that's kind of all I could hope for.